Hey everyone. Hey everyone, welcome to the live class. We'll wait 2 more minutes and then I'll start. If you can see the board and my voice is clear, please say yes in the chat. All right, that's good. Okay, so let's start the class now. Today's chapter is about A2 biologies. Now, for the IMAT, again, you need to know the AS and A2 syllabus. And this chapter is about respiration. And this chapter is very popular in both the IMAT and A level. In all of the IMAT exams, you will find a lot of questions regarding respiration. So what is this chapter about? It's about energy. So basically, how energy is produced, how we make energy, what is mitochondria, all of this stuff is discussed in this chapter. Now, energy, what is energy? ATP is the universal energy currency as it provides the immediate source of energy for cellular purposes. Now, in our body, there are two types of reactions. There is the anabolic reactions, which require energy. So we need energy to make anabolic reactions, right? And catabolic reactions are the reactions that release energy. So in order for anabolic reactions to happen, we need catabolic reactions. Anabolic reactions are basically the reactions like muscle contraction or protein synthesis or DNA replication. So all of this stuff is anabolic reactions. And catabolic reactions, which releases energy, is basically when ATP is produced. So ATP production is catabolic reaction because ATP releases energy. So let me just zoom in. Right. Now, ATP is the universal energy currency as it provides the immediate source of energy for cellular purposes. So ATP is the molecule that is the source of energy in our body, and it is needed in living organisms. Again, it's an anabolic reaction. For anabolic reactions, we need ATP for anabolic reactions. Reactions like DNA replication, active transport, movement, maintenance of our constant body temperature, which is 37 degrees. So all of these reactions require energy or energy in the form of ATP. Now, what is ATP? ATP is adenosine triphosphate. You don't need to know the name, but what you need to know is it's a universal energy currency and it has high turnover for energy. So the reason that our body produces ATP for energy is because ATP is a small molecule and it's water soluble and it's readily hydrolyzed to release energy. So when we take food, right, that food is converted to glucose. Now, you might ask, why isn't glucose used instead of ATP? Because ATP is water soluble, it's small, it is readily hydrolyzed to release energy, whereas glucose is a big molecule. So suppose this is glucose, this is ATP. So ATP is much easier to transport. Again, all of these things help ATP to be a better energy currency or energy source. Now, how is ATP synthesized? Well, let me just move this here. Yeah. So ATP is produced during respiration, and this happens in substrate-linked reactions. What is this? I'll explain in a moment, but this is the structure of ATP. So it has an adenine group, a ribose sugar, and this right here are 3 phosphate groups. So these are 3 phosphate groups, right? And when phosphate groups are released, right? So when, let's say, this phosphate is released, when it's released, energy is released as well. So going into coenzymes now. What is a coenzyme? A coenzyme is basically a hydrogen carrier molecule. What do I mean by hydrogen carrier molecules? Hydrogen carrier molecules will accept hydrogens, and hydrogens are needed for making ATP. I'll also explain this later on. So now there are 3 different coenzymes you need to know. It's NAD, FAD, and there is one more. It's called NADP. This coenzyme will not be used in mitochondria. This will be used in chloroplast. So you don't need to know a lot about this right now, but there are 2 coenzymes in our body which are needed for respiration. These are NAD and FAD. You don't need to know the names. So coenzyme A is another coenzyme, and its job is to carry acetyl groups into the Krebs cycle. Again, this is not important. I'll explain this more in later on. So now let's get to the main part. That was just the starting. Now comes the main part, which is respiration. Respiration is the process where energy from complex organic molecules like glucose or lipids is transferred to ATP. And there are 2 types of respiration. There is the aerobic respiration and the anaerobic respiration. Anaerobic respiration, you've learned it earlier. It's when you respire in the absence of O2. So O2 is not present. Oxygen is not present in anaerobic respiration. But in aerobic respiration, you have oxygen. So aerobic respiration has 4 different steps. There is glycolysis, which happens in the cytoplasm, link reaction, which happens in the matrix of the mitochondria. Krebs cycle also happens in the matrix of the mitochondria. And then the last stage, right, which is the most important one, oxidative phosphorylation. This is the place where oxygen will come in play. And oxidative phosphorylation happens in the inner membrane of the mitochondria. So why are respiration divided into these steps? Well, there are 3 reasons for this, because the first one is it allows precise control of energy released. So this is the first reason why we divide aerobic respiration into 4 stages instead of one stage where we can just take glucose and convert it into ATP. Instead of doing this, we divide aerobic respiration into 4 steps. This is one of the reasons. The second reason is that our cells cannot harness the total energy in one step. So total energy cannot be harnessed in one step. That's another reason. And the third reason is that glucose is a big molecule and it cannot be translated to ATP or transformed to ATP in one simple step. So let's start with glycolysis first. Glycolysis is the first step of converting glucose to ATP. So and remember, this happens in the cytoplasm. This is very important. Cytoplasm. Glucose happens in cytoplasm. So, glycolysis happens in cytoplasm. So we have the glucose, right? We get the glucose, which is 6-carbon. So it's a 6-carbon molecule. And we get ATP. ATP is already present in the body. So this ATP will convert glucose into fructose phosphate. What is fructose phosphate? Basically, we have 6 carbon molecules, right? 1, 2, 3, 4, 5, 6. Ignore this. So what ATP does to this glucose is it adds 2 phosphate groups to each end of the 6-carbon chain. So this becomes fructose phosphate. And then fructose phosphate becomes fructose biphosphate. Again, ATP is needed here. So fructose biphosphate is made, and then fructose biphosphate will be converted to 2 different molecules. So initially there was 6 carbon atoms. Now there would be 3 carbon atoms each in each molecule. So this fructose biphosphate becomes something called triose phosphate. So each of this molecule is called triose phosphate. It has 3 carbon atoms, each has 3 carbon atoms. And after triose phosphate is made, we use something called NAD. I explained you earlier what NAD is. NAD is a hydrogen carrier. So NAD, there are 2 NADs in this reaction. Glycolysis uses 2 NAD. So NAD will gain a hydrogen because remember, NAD is hydrogen acceptor. Coenzymes are hydrogen acceptors. So NAD plus hydrogen becomes something called reduced NAD. Reduced NAD can also be written like this, NADH, where this H basically means that the NAD gained 1 hydrogen. Since there is 2 NADs, 2 hydrogens will combine with 2 NADs to make 2 NADH, right? 2 reduced NAD. Now, when this happens, triose phosphate will become an intermediate. What is this intermediate? You don't need to know, but what you need to know is that this intermediate will become something called pyruvate. And when it becomes pyruvate, 2 ATP molecules would be released. So this pyruvate, when pyruvate is formed, it releases 2 ATP. Now, why does all of this happen? Because of this ATP right here. So in glycolysis, right? In glycolysis, what do we get? We get 2 ATP and 2 reduced, or simply I can write NADH. I get 2 ATP and 2 NADH. I'm making 4 ATPs in this entire molecule, in this entire reaction. So glycolysis makes 4 ATP, but 2 ATP are used right here. So I use 1 ATP when I convert glucose into fructose biphosphate, and then I use another ATP molecule when I convert fructose phosphate to fructose biphosphate. So in total, I make 4 ATP in glycolysis, but since I use 2 ATP in the process, I'm making a net gain of 2 ATP. So in glycolysis, I make 2 ATP, 2 reduced NAD, and 2 molecules of pyruvate. So these 3 carbon molecules will become pyruvate. And again, this means that pyruvates are 3 carbon long. Now, in summary, what is glycolysis? Glycolysis is the phosphorylation of glucose and the splitting of fructose biphosphate into 2 triose phosphate molecules, which then further oxidize to pyruvate with a small yield of ATP and reduced NAD produced. So 2 reduced NAD, 2 ATP, and 2 pyruvates are produced in glycolysis. Now, you also need to know what phosphorylation of glucose is. Phosphorylation of glucose happens in step number 1, where glucose becomes fructose phosphate. This is called phosphorylation, and phosphorylation is addition of a phosphate group. So addition of phosphate means phosphorylation. Now, this was the first step of respiration, where you produce pyruvate, 2 molecules of pyruvate. Now, this pyruvate, right, 2 molecules of pyruvate will go from the cytoplasm, because remember, glycolysis, I told you earlier, happens at the cytoplasm. So the pyruvates that are produced in the cytoplasm would then move out from the cytoplasm into the mitochondrial matrix, because I told you in somewhere right here, wait, yes, right here, that glycolysis happens at the cytoplasm, but link reaction will happen in the mitochondria, more specifically in the matrix of the mitochondria. So this link reaction happens in the matrix of the mitochondria. So pyruvate will go into the matrix via active transport. This active transport will require ATP, because active transport needs energy. So pyruvate goes from cytoplasm to mitochondrial matrix. And what does pyruvate become now? Because in link reaction, the pyruvate will undergo decarboxylation and dehydrogenation. So this step is called decarboxylation. And basically, this step is when CO2 will diffuse out of the cell, basically decarboxylize. Pyruvate will decarboxylize. Pyruvate would release one CO2 molecule. And when it does, right, so this pyruvate now will release one CO2 molecule, and it would become 2 carbon chain long, because 1 carbon is released in the form of CO2. Similarly, the same thing happens with the second molecule, because there are 2 molecules of pyruvate, produced in step 1, which will go to the step 2 or link reaction. So I make 2 molecules of carbon now instead of 3. CO2 is released in the second pyruvate molecule as well. And then what happens is these 2 pyruvate molecules now will release a hydrogen. So each pyruvate molecule releases a hydrogen. This step is called dehydrogenation or dehydrogenase. So when these 2 hydrogens are produced, these 2 hydrogens would combine with NAD. So 2 NAD plus 2 hydrogen molecules will give you 2 NADH, or you can say NADH is reduced NAD. And what happens next is this molecule now, these two molecules, when combine with something called coenzyme A. What is coenzyme A? This CoA is a carrier molecule, which will carry pyruvate into step 3. So yeah, it's very difficult because last time I was using a different whiteboard. So pyruvate will combine with coenzyme A to make acetyl coenzyme A. So what are you making in the second step in link reaction? In link reaction, you are making two molecules of acetyl coenzyme A, and you are also making two NADH molecules. And you are also making two CO2 molecules, because remember, two CO2 molecules were released in this part right here. So link reaction makes two acetyl coenzyme A, two NADH, and two CO2, right? Basically, this is the summary of link reaction, where pyruvate would release two hydrogens. So NAD would gain the two hydrogens and become reduced NADH. And you will also release two carbon dioxide molecules. And what you get is acetyl coenzyme A, which is basically pyruvate combined with coenzyme A. So acetyl coenzyme A is pyruvate plus coenzyme A. So when you combine these two, you make acetyl coenzyme A. Now this acetyl coenzyme A is carried to step number 3 of the respiration, which is step 3 is called the Krebs cycle. Krebs cycle is happening in mitochondria, as I explained earlier. Where it is? Yeah, here. Krebs cycle happens in the mitochondria, and more specifically in the matrix. So these two steps right here happens in the mitochondrial matrix. All right. Now, what is Krebs cycle? Krebs cycle is basically this acetyl coenzyme A combining with a molecule called oxalacetate, which is 4 carbons long. Now, Krebs cycle has 7 steps, right? You can learn this cycle right here, or I'll explain each cycle differently. So the first step is that this acetyl coenzyme A will combine with a molecule called oxalacetate. And this oxalacetate is 4 carbons long. And acetyl coenzyme A is 2 carbons long. So when these two combine, you get a 6 carbon molecule called citrate. So in Krebs cycle, step 1 is you make citrate. Step 2 is this citrate will release carbon dioxide. So citrate will release CO2 and a hydrogen. This hydrogen will combine with NADH. It will combine with NAD and make NADH. So step 2 is you make CO2 and NADH. Step 3 is again this citrate will release two hydrogen, one carbon dioxide and NADH. So this citrate will release one hydrogen. This hydrogen will combine with NAD and make NADH, and you'll also release carbon dioxide. Now, before I continue, I told you earlier that acetyl coenzyme A are two molecules, because remember, for each respiration, I'm making 2 molecules of pyruvate. So 2 molecules of pyruvate make 2 molecules of acetyl coenzyme A. 2 molecules of acetyl coenzyme A make 2 molecules of citrate. So right here, I have 2 citrate molecules. So whatever is happening, right, whatever is being produced will multiply by 2 because there are 2 molecules in each step. So 2 molecules of citrate will make 2 molecules of CO2 and 2 molecules of NADH. Again, step 3 also makes 2 molecules of CO2 and 2 molecules of NADH. Step 4 is called ATP. So ATP is released in this step. And since there is 2 molecules of citrate, each citrate molecule will make 2 molecules of ATP. Step 5 is the citrate. This citrate would release a hydrogen, which will combine with another molecule. This molecule is called FAD. I explained FAD earlier at the start. So I told you there is FAD and NAD in the mitochondria. Both of these are hydrogen acceptors. So citrate will release a hydrogen. This hydrogen will combine with FAD to make FADH or reduced FAD. So both FAD and NAD have the same function. They accept hydrogens. So citrate accepts hydrogen in step number 5. And step number 6 is you make intermediate. You don't need to know the name of the intermediate. Step 7 is this intermediate will release another hydrogen to make NADH. So step 7 is you make NADH or reduced NAD. So in total, there are 7 steps of Krebs cycle. Step 1 is you make citrate, 2 molecules of citrate. Step 2 is you make 2 molecules of CO2, 2 molecules of NADH. Step 3 is you make 2 molecules of CO2 again and 2 molecules of NADH again. Step 4, you make 2 molecules of ATP. Step 5, you make 2 molecules of FADH. Step 6 is you make 2 molecules of NADH again. Step 7, you make 2 molecules of NADH. Sorry, this step 6 is when intermediate is formed. So in step 6, you're basically doing nothing. This intermediate will go into step 7 and make NADH, which is also 2 because there is 2 molecules of citrate. So I'll till now, how many ATP, CO2, NADH, FADH did we make? In glycolysis, right? We made 4 ATP. So 4 ATP were produced. But since 2 ATP is used, I make a net ATP of 2. So in glycolysis, I make 2 molecules of ATP. And were there any CO2 being produced in glycolysis? No. So 0 CO2 produced in glycolysis, and 2 molecules of NADH were produced in glycolysis. 0 molecules of FADH were produced. In link reaction, did I make any ATP? I did not. So 0 molecules of ATP were produced in link reaction. 2 molecules of CO2 and 2 molecules of NADH were produced in link reaction. In Krebs cycle, I'll go back to Krebs cycle. In Krebs cycle, what I made was 2 molecules of ATP. So 2 molecules of ATP in step number 4 and 4 molecules of carbon dioxide, 4 molecules of carbon dioxide, 2 in step number 2 and 2 in step number 3 to make total of 4 carbon dioxides. And I also make 6 molecules of NADH, 6 molecules of NADH, 2 in step number 2, 2 in step number 3, and 2 in the last step. Step number 7 makes 2 molecules of NADH. So in total, there were 6 molecules of NADH. And I also made 2 molecules of FADH or reduced FAD. So there's a question. So we do not make nor use ATP in link reaction. Yes, we do not make or use ATP in the link reaction. What link reaction is, why link reaction happens, it's because of NADH. We need NADH for the last step, which is oxidative phosphorylation. It will all get more clear when I get to this, but the link reaction, the purpose of link reaction is to make NADH, not ATP. So when all of this happens, right, all of this happens, step 3, when step 3 finishes, Krebs cycle finishes, I go and move on to step number 4. Now, this step is the most important of the step. And questions are usually asked from this last step, which is oxidative phosphorylation. Now, what is oxidative phosphorylation? Oxidative phosphorylation is when all of these NADH, right? All of these NADH are taken from the mitochondrial matrix to the place where oxidative phosphorylation is happening. So the NADH that are produced will go on to the place where oxidative phosphorylation happens. And I explained earlier, where is it? Yeah, here. Oxidative phosphorylation will happen in the inner membrane of the mitochondria. You'll also learn what the different types of structure of mitochondria. But for now, oxidative phosphorylation happens at the inner membrane of the mitochondria. So what is oxidative phosphorylation? Now, the hydrogens that were from the NADH, right? NAD and FAD would release hydrogen. So these NADH and FADH would release hydrogens and they would become NAD and FAD respectively. So each NADH will release one hydrogen, and when this hydrogen is released, NADH would become NAD. So NAD can go back to glycolysis and start respiration again. So these NADs are being recycled constantly. And similarly, FAD will release 2 molecules of hydrogen. And this FADH can carry 2 molecules. FAD can basically carry 2 molecules of hydrogen. NAD can only carry 1 hydrogen molecule. So when FAD, a reduced FAD or FADH will release 2 hydrogens, it will make FAD, and FAD will go back to Krebs cycle and gain hydrogen again. So what happens when these hydrogens are released? So these hydrogens, right, would further break down or split and would make protons and electron. What is a hydrogen molecule? Hydrogen molecules basically have 1 proton and 1 electron, right? 1 proton and 1 electron. So 1 proton and 1 electron is released in NADH. When NADH becomes NAD, 1 proton and 1 electron is released. When FADH becomes FAD, so reduced FAD becomes FAD, and it releases 2 hydrogen molecules, there would be 2 protons released and 2 molecules of electrons released. Why am I explaining this? Because of this step right here. So let me just clear everything now. Because this is the most important part. So the reduced NAD would release hydrogen. The hydrogen becomes protons and electrons, right? And these protons and electrons would go to the something called the electron transmission chain. Now, these electrons that are produced will go through this electron transmission chain, and these electrons are energetic. So energetic electrons releases energy as they pass on through the electron transmission chain. So what is electron transmission chain? It's basically the place where electrons release energy while they move. And electrons will go from the matrix to the intermembrane space. And this energy from the electrons are used to transport these protons, right? These protons from the matrix, from the mitochondrial membrane, from the matrix to the intermembrane space. So the protons move from the mitochondrial matrix to the intermembrane space, and protons then return back to the mitochondrial matrix. So this step right here is when protons move from the matrix to the intermembrane, right? This is where these protons will move from the matrix to the intermembrane space. And then these protons, this happens because of the energy released by the electrons. So this process where protons move from the matrix to the intermembrane space is because of electrons releasing energy. And then these protons will go back to the intermembrane, to the mitochondrial matrix. So first step, protons move from the matrix to the intermembrane space. In step 2, the protons move back from the intermembrane space back to the matrix. And why do they move back? Well, because of something called ATP synthase, right? This protein structure right here is called ATP synthase. Now, what is this structure? Well, ATP synthase is basically a protein that will synthesize ATP. So it's a coenzyme, right? And when protons move from the intermembrane space to the matrix, the ATP will be synthesized. So the ATP synthase rotates when protons move to the matrix. When protons move from the intermembrane space to the matrix through the ATP synthase, ATP is released. So proteins return to matrix via the ATP synthase, and this provides energy for ATP synthesis. So the movement, right, where the protons move from the intermembrane to the matrix, this causes ATP production. ATP is synthesized right here, and ATP is synthesized, and then when this proton goes to the matrix, right? When it goes to the matrix, this is where oxygen comes into play. Oxygen will combine with electrons and hydrogens to make water. So oxygen, O2, would combine with the protons, 4 of these protons, in fact, and 4 of the electrons, which were in the electron transmission chain, to make 2 molecules of H2O, right? What is the role of oxygen in aerobic respiration? It will attach with the protons and the electrons to make 2 H2O, or basically 2 molecules of water. And in all of this process, where ATP is produced, right, there is 28 molecules of ATP being produced. So oxidative phosphorylation is the biggest production of ATP. So when I go to this table right here, oxidative phosphorylation makes 28 ATP molecules. Glycolysis makes 4, link reaction makes 0, Krebs cycle makes 2 molecules of ATP. So the biggest production of ATP happens at the oxidative phosphorylation. And in total, in all of this process, I'm making 32 molecules of ATP, 28 in oxidative phosphorylation, 2 in glycolysis, and 2 in Krebs cycle. In total, there is 32 molecules of ATP. that I get. So this was basically respiration. Now, I'll do this in the next class because I'm running out of time, I think. And let's do this question now. This is from, I think, I don't remember the IMAT exam, but I'll give you guys 2 minutes to solve this question. In which of the following stages of mammalian respiration is CO2 released? In glycolysis, anaerobic respiration, or Krebs cycle? I'll give you guys 2 minutes. Actually, let's do anaerobic respiration. I'll do this question in Discord. I'll send it on Discord if we run out of time, but let me explain anaerobic respiration. Where is anaerobic respiration? So